Hi, and welcome to my poster presentation. Today, I'd like to talk about the role of the seemingly innocent substrate in dioxygen activation at Rieske Dioxygenases. My name is Katja Chisi, and we've been working on this project in a collaboration between the Reier Group and the Hofstetter Group at ETH Zurich and EABAC. Rieske Dioxygenases are a subclass of metalloenzymes that perform Cis-Dihydroxylation of aromatic hydrocarbons with molecular dioxygen. In organic chemistry, O2 reactivity is kinetically hindered, which is why activation generally proceeds through the generation of metal oxygen intermediates. The active sites of Rieske dioxygenases indeed contain such a metal site, which is, however, in its resting state entirely unreactive towards O2. The generation of these reactive species proceeds through three main steps, including reduction via an electron, release of a water ligand creating an open site, and coordination of the substrate. This latter step is in particular interesting because the substrate itself does not bind or coordinate to the metal site, but is instead located nearby within the active site pocket. The question that comes up is how does a seemingly innocent and non-interactive substrate can control dioxygen activation at a transition metal site? As theoretical chemists, we designed a theoretical approach to answer this question, which consists of three main steps. First, we generate a local model including only relevant structural entities of the active site, which we can describe in a quantum chemical manner. Second, we expand this picture through inclusion of the protein backbone, which can be modeled through a force field. And ultimately, we want to map our findings of these first two steps onto different representatives of this enzyme class, which should be screened in an automated way. Today I want to take a closer look at the first step. This minimal model is not only computationally feasible, but the neglect of backbone effects allows us to understand the underlying chemistry in an isolated fashion. However, as effects of the environment can be crucial, the choice of this local model is not straightforward and very sensitive. We performed a quantum mechanical study on the active site of naphthalene dioxygenase, which is a model representative of Rieske dioxygenases. To exploit the previously mentioned advantages of our local model and also account for its difficulties, we first generated multiple structural active site models, where for each we sampled a space of chemical reactions, including all relevant elementary steps on the path to dioxygen activation and sequences thereof. For each elementary step, we tested a suite of computational methods. Overall, this yields us a very large dataset of reaction energies and properties for the respective elementary steps, from which we can extract contributions of the substrate to dioxygen activation. From this dataset, we find three main characteristics. First, release of a solvent-derived water ligand seems to be largely insensitive to the presence of the substrate and appears to be a thermoneutral process. Second, presence of dioxygen prior to electron transfer does not lead to formation of any of the known reactive iron-oxygen species. And only after reduction, we find consistent evidence across the suite of computational methods for contributions of the substrate to dioxygen activation. Precisely, the additional electron in the active site is found as a resonance-stabilized charge at the substrate instead of the metal site, narrowing down that the presence of the substrate is critically coupled to this electron transfer step. With this, I want to thank you for your attention.